All right, so this is part 2 to chapter 2. We're going to go through the eukaryotic cell. This should again be review, but I'll be going through the organelles for you. So this is a eukaryotic cell. Eukaryotic cells are characterized by a membrane-bound nucleus, membrane-bound organelles on the inside, as well as being larger and more complicated or more complex than a prokaryotic cell. So let's move to our features of a eukaryotic cell, starting with the plasma membrane, or letter B in your outline. The plasma membrane, as you see here, is an organelle that's made of many different molecule types. So the fluid mosaic model is the term you use and hear a lot with the plasma membrane. Fluid refers to the fact that it is in constant motion. So these phospholipids are moving past each other, they're in motion and fluid. The proteins are also mobile, so unless they're locked in place by being anchored down to the cytoskeleton, they're allowed to move laterally in the membrane. So these guys can move around, and so that fluid nature of the molecules is where fluid comes from. Mosaic, right, refers to the fact that there's many types of molecules that create the whole membrane. So we have primarily a phospholipid bilayer. All right, so this is our primary sort of backbone of our membrane is a bilayer of phospholipids. Our heads are going to face the external environment because it's watery. The inside of the cell is also watery, so the heads on the second layer are going to face inside the watery compartment of the cytoplasm or the cytosol. And inside, right, we have a sandwich of fatty acids, and this is going to be a non-polar component, so we have to think about this when we think about substances that can cross through the cell, either in or out through the membrane. So the mosaic, the first part that creates this mosaic is phospholipids. I mentioned in the outline that some phospholipids may have a carbohydrate bound to it, so you can call that a glycolipid. Moving on to our proteins. So proteins are another major component of our cell membranes. Proteins that thread all the way through or are both outside and form some of the interior are called integral membrane proteins. They integrated themselves into the membrane. Whereas some proteins associate with only one side, and you call those peripheral proteins, right? This guy over here. Some proteins will have carbohydrates bound to them, so we call them glycoproteins. All right, so in the notes, these integral proteins are also called transmembrane proteins. All right, coming back here, also transmembrane. Some proteins will form a channel through the center, so it'll be kind of a hollow tube or tunnel through them, and we can call them channel proteins. We'll see those later on in the lecture. And then lastly, we have our cholesterol. The cholesterol molecules, as you see here in these yellow molecules, are embedded with the lipid layer of our membrane. So remember, cholesterol is a lipid. These steroid molecules are lipids, and so they are going to naturally fit within this lipid bilayer within the fatty acid tails. The cholesterol that's there is going to maintain the fluidity of the membrane. So the membrane, the fluidity of the membrane, or how fast and slow the molecules move, is critical to appropriate membrane function or cell function. And these molecules act as antifreeze. So they are going to, if the cell starts to become too rigid and the molecules are not moving enough, the cholesterol there can start to loosen them up, or if it's going too fast, the cholesterol can slow it down. So it just mediates the fluidity. Moving into the cell, okay. This is a ligand-gated channel. So I do talk about channel proteins. Let's go back up to proteins, forgot to mention this. The channel proteins will form an opening, right? So the proteins can allow substances to go in or out through an internal channel. But sometimes these channels are gated, which means that they're closed most of the time and only open up when there is a substance that binds to the channel to help it open. So a gated channel is the term we used for when it's closed most of the time, but then some stimulus opens it up. So so we have different stimuli. We can have an electrical charge, we can have open up the channels, or we can have a ligand. So the word ligand is any molecule that binds to a protein, that loosely associates with a protein. So sometimes a molecule, in this case, in this example, we have a neurotransmitter. So the neurotransmitter, a ligand, is required to open this ion channel. So this is acetylcholine. Acetylcholine, when it binds, you see the binding here, it binds to the receptor and it opens up the channel so that we can have the ions moving in response through the channel. Okay, so that's just an example of a gated channel, and we'll look at these ligand-gated channels later. All right, so what do you think the answer is to this? I'll pause the lecture and think about your answer. Okay, so the answer is going to be nonpolar. Okay, so we're now here and here, because remember the interior, right? So here's our phospholipid heads, and the interior, the middle part of our membrane, is really nonpolar, right? So it's kind of like the oil in water and oil. So molecules that can move through the membrane have to be able to move through the nonpolar layer. So we're at B or D. So nonpolar, and therefore are they hydrophilic or lipophobic molecules, or is it hydrophobic and lipophilic molecules? Your answer is letter D. Hydrophobic is correct. This is a nonpolar molecule is hydrophobic. It does not dissolve in water. Lipophilic means loving of lipids, right? Lipo refers to lipids. Philic means to love. So this is an attraction to lipids and a repulsion of water. So letter D. All right, great. So we're going to move through the different organelles. I skipped the cytosol. Let's just go back and say a couple words about the cytosol. So this is the internal environment of the cell. It is mainly water, but it's a viscous material. So the cytosol is thick, kind of like a jelly, and it contains a very high concentration of ions, nutrients, dissolved and suspended proteins, enzymes. A lot of the chemical reactions that occur inside the cell occur in the cytoplasm. Okay, let's move on to our nucleus. So remembering that the nucleus is a the nuclear envelope is a double membrane, so we can see the two layers here. The two layers will come together to form pores. Those nuclear pores will allow the transport of substances into the nucleus for gene expression. RNA molecules need to move out, again, for translation. So that's what the pores are for. The nucleolus is a dark, staining, dense region within the nucleus that creates ribosomal RNA. So this has a function to make ribosomal RNA. And then chromatin is the loose, barely there. You can kind of see this is kind of making like little lines in the background. Chromatin is loosely packed DNA. Okay, so if the cell is not dividing, the DNA is in chromatin form. When the cell needs to undergo mitosis, it will condense its chromatin into chromosomes. So you don't see chromosomes unless the cell is actively dividing. All right, moving on to the ribosome. All right, this is just a pretty picture of our nucleus. So we can see all those really cool nuclear pores. The next organelle is the ribosome. So ribosomes are these really tiny protein, they're always depicted as these little circles. The ribosome itself, though, is, I don't have a picture, oh, here it is. This is our ribosome. It has a large subunit and a smaller subunit that sit on top of each other, And since you've had biology, you know that the messenger RNA will be sandwiched between those subunits and we can have translation occur. So this is a site of protein synthesis, right? We have translation happening and protein synthesis. The other molecule that creates the ribosome is our ribosomal RNA. But in terms of where you find the ribosome is found both on a membrane, the rough ER, okay, or it can be free in the cytoplasm, which we call free ribosomes. So the ribosomes that are on the ER, so the rough endoplasmic reticulum, the proteins made here are mainly to be shipped out of the cell. Shipped out of the cell. Okay, so I'll put protein. And the proteins that are created on these free ribosomes usually stay in the cell. Okay, so we just have two different fates for our proteins. All right, so those are the ribosomes for protein synthesis. Let's move on to the endoplasmic reticulum. So sorry, I'm just scrolling through my pictures here. The endoplasmic reticulum is going to be a network of folded membrane that is continuous with the nuclear envelope. So take a look at what's going on in this circle. Notice that the purple nuclear envelope, right, becomes the blue ER and just extends away from it and forms all these convoluted compartments. So the ER is actually an extension of the nuclear envelope. And notice that the smooth ER is also an extension. It's not actually depicted very well here, but it's an extension of that same material. Actually, you can see some smooth ER right here. So the difference between smooth ER and rough ER is the ribosome, right? The ribosomes on the rough ER make that rough ER a organelle for protein synthesis and shipment. Whereas smooth ER, since it does not have those ribosomes, it's going to have different functions. And the functions for smooth ER are lipid synthesis, right? So the lipids we talked about already are prostaglandins, which is our eicosanoids, our fats, our phospholipids, steroids. These can be synthesized in the smooth ER. And as well as detoxification. So the smooth ER can detoxify molecules in the cell. All right, so the next topic here would be the Golgi apparatus. Now, substances created in the endoplasmic reticulum, or the ER, protein in the rough ER, lipids in the smooth ER, they're going to travel to the Golgi apparatus for modification and for sorting and shipment. So the vesicles are going to bud off of the ER and travel to the Golgi apparatus and come to the Golgi apparatus at the cis face. So it's a unidirectional. So these little guys here, these are vesicles, and they're going to contain the molecules that are newly synthesized either from the smooth ER or the rough ER. So they're going to come into the Golgi apparatus. The vesicles will fuse with the Golgi apparatus membrane and then put in, right, deliver the molecules into the Golgi apparatus sacs. Now, the Golgi apparatus is, in this picture, we have 1, 2, 3, 4, 5. It's a series of about 5 flattened sacs. I like to call them pita bread, or think about them as pita bread, because they're hollow, but they're one unit, right? So it's one pita bread, 5 of them stacked together. And these molecules will move through from one pita bread to the next, to the next, to the next, all the while getting modified, and then eventually leave off the transface by budding into a vesicle. And those vesicles can be used to transport those substances to the plasma membrane for secretion. So this fusion here with the plasma membrane, then the substances inside through exocytosis can be liberated from the cell. So the Golgi apparatus is a place that modifies your substances created by the cell, and it can pack them up together and then ship them to the appropriate area of the cell. So maybe they're going out of the cell, maybe they're not. Maybe they're going to another organelle. All right. Another job of the Golgi apparatus is that lysosomes are created here. Here's our lysosome picture. So lysosomes are pretty boring. They're just, to look at, right? They're just a circle. They're a membrane-bound sac, but inside the lysosome is really important chemical reactions. I'm flipping through to see if I have another picture, but I don't. So lysosome is going to have hydrolytic enzymes and a low pH. So an acidic pH, and it's also going to have enzymes. And this is going to be an organelle that will fuse with a larger vesicle. So here's our lysosome. Let's say that our cells have brought in some kind of large particle, such as a bacterium. Let's say that a bacteria has been caught by our white blood cell, and we want to destroy this bacteria. So the enzyme, the lysosome, will fuse with this vesicle that brings in the bacteria. Actually, it's called a vacuole at this point. The lysosome will fuse, and then with that fusion, the enzymes and the acids will come in and digest this bacteria, but in a very neat way, right? Because we are doing this digestion within a membrane-bound compartment, completely surrounded by membrane, so that acid and those hydrolytic enzymes are not going to accidentally digest anything else that the cell wants. It's contained within that vesicle. All right, so peroxisomes are very similar to lysosomes, and peroxisomes will contain hydrogen peroxide, which is this, and hydrogen peroxide can destroy, detoxify and destroy other particles inside the cell, but hydrogen peroxide is actually kind of a nasty substance, so hydrogen peroxide can be broken down by an enzyme to water and oxygen. All right, so let's move on to our mitochondria. So our mitochondria is going to be a double membraned organelle. We have an outer membrane and an inner membrane. We'll talk about this in the cell respiration. This space between the membranes is called the intermembrane space, and the inner membrane is folded. It's very convoluted, and those folds are called the cristae. And internally, the most interior compartment of the mitochondria is called the matrix. And many of you know that the mitochondria is for ATP production, which is the energy of our cell. We'll go over that again. And in the notes I have that the mitochondria has its separate DNA. So the DNA that's found in the mitochondria is called mitochondrial DNA. It is has information that can create another mitochondrion. So if the cell, it needs more energy, it can actually, the mitochondria can reproduce itself, have more mitochondria within the cell, and that for the ATP, you can have more ATP to use, right? Because you have more mitochondria making those ATP. All right, let's move on to, so I already talked about peroxisomes. This is the oxidases can neutralize free radicals, and peroxisomes can also detoxify alcohol. The next topic here would be the cytoskeleton. So the cytoskeleton are a network of protein fibers. So these are going to be filaments. Remember, we talked about how proteins can form long strands, and those are filaments. So we have different forms of these protein fibers. They are microtubules, which are tubes. They are, so here's a picture of a microtubule. And if you look carefully, it is pictured like a tube. Then we have intermediate filaments that are going to be intermediate in size, microtubules being the largest. And then microfilaments are the smallest. So they all have different sub-jobs, but overall, the cytoskeleton are to create the shape of the cell, so maintain the entire shape of the cell. give the cell its unique scaffolding and shape. The cytoskeletal fibers, as you can see, can bind to your organelles to stabilize them in place, to hold them as a sort of a structural component, to hold them and to give them stability inside the cell. The cytoskeleton can form structures like cilia and flagella, which we'll look at, as well as their dynamics. So you want to think about the cytoskeleton as kind of like these filaments that can build, they can elongate and shorten at will. So if the cell needed to change its shape, such, for example, you have a white blood cell that is able to crawl, right? So if it's a mobile cell and you wanted to crawl from one place to another, it can actually push out the the plasma membrane, right? Push the cell in one direction, and this is by building up the cytoskeleton in one direction, and then you're breaking down in the back. You're breaking that down. And then over lots and lots of building up and breaking down, right, we can get these overall directional movement of the cell. So it actually can move the cell. Cells that are mobile will have to rearrange their cytoskeleton to move. Here are microvilli. So microvilli are made of these small filaments, these microfilaments, and they are basically these projections of the cell membrane. You can see how tiny they are. If I were to draw this, right, this cell membrane, you would have this upward projection and then it comes down. And the reason why they're held in place here is because we have tiny fibers, these microfilaments, holding the shape of each individual microvilli. The reason why we have microvilli on the surface of some of our epithelial cells is to increase our surface area. Okay, I'm sure if you took me for anatomy, you've heard this a lot, right? So the surfaces of cells that need to secrete or absorb a lot will have microvilli, so that the surface area, right, the amount of area that's in contact with the outside is increased, so that you can have more absorptive surfaces, right? You can absorb things through the flat area of the membrane. You can also absorb through all of this part, this upward projection, the interior. So a lot of absorbing can happen. Here is just a larger, nicer picture of the two. So this is a cool picture of two cells. You can see there's a mitochondria right there. All right, let's recap with a quick quiz. Where is ribosomal RNA synthesized? Answer is nucleolus. And then here is just a rundown of all of the organelles that we just went through very quickly. And this is something that's good to look at and maybe have blank and fill it in. But this just covers everything. The plasma membrane, the nucleus, nucleolus, and go on down, lysosomes, peroxisomes. I didn't talk about centrioles, so let me just talk about centrioles real quick, which is also in your outline. Centrioles are going to look like these. Let me find a picture of this cell. I think we have to go back to the very first one we looked at. So here's our centrioles. Okay, this should look familiar to you. These are very important in mitosis and meiosis, because that is where spindle fibers will grow from, and they're going to allow the movement of the chromosomes. So these centrioles are going to organize, set up the poles during mitosis. They're going to grow the spindle fibers, and the spindle fibers are therefore structures that move physically the chromosomes during cell division. All right, let's move back to our slide where we were, the table. All right, and then here's our cytoskeleton. So quick review of our organelles of a eukaryotic cell. And then we're going to talk about the next topic, which is our intercellular attachment. So if we want to attach two cells together, cells do this by proteins. The proteins used in these junctions are called cell adhesion molecules, or CAMs. They are going to be proteins, they're transmembrane proteins, and they form different kinds of junctions. So different kinds of CAMs for different kinds of junctions. So let's look at a few. So looking at this is actually a picture of a tight junction. This is a desmosome. Let me see here. I thought I had a picture of a gap junction. Oh, no, okay. Here are the gap junctions. Sorry. So I start with the gap junctions in your outline. Communicating junctions or gap junctions are where two cells are bound together, but the junctions between them are hollow. So we have a channel that's created by these cell adhesion molecules. And this allows for materials in one cell to quickly go into the next cell, right? So we can communicate chemically between two cells with the, whatever the ions or the chemicals are. In this way, we can coordinate the message from one cell to another cell, coordinate movement. So like cilia beating can be coordinated for cells that have gap junctions. The cardiac muscle cells have gap junctions so that it can coordinate the heartbeat more easier, as well as smooth muscles have that same gap junctions to coordinate contraction. The next kind of junction is called an adhering junction or tight junction. So here is, let's go back to this one. This is going to be our tight junction. And you can see how the proteins, the cell adhesion molecules are different. And they literally button up the two cells side by side. And when you do this, so in this picture here, we have the bloodstream, we have some interstitial fluid, and then we have the lumen of an organ. So this could be maybe your small intestine. And notice how the cells of your small intestine are tightly bound together, right? So because we have these tight junctions zipping up and forming a very tight very tight seal between those two cells, it's going to make it very, very hard for substances to go through those cells into the interstitial fluid and into the bloodstream. So it really controls what goes in through this layer of epithelial cells, and it allows the epithelial cells to be the control mechanism of what molecules move through that membrane, right? Because that little space between the cells is sealed off with tight junctions. So if you look at adhering junctions or tight junctions in the notes, they're going to prevent the permeation of substances through, seal off any space between cells, and prevent movement of water or other molecules past. So even water cannot fit through those tight junctions. You'll find junctions like this on the lateral sides. So you can see that if we have this is one kind of square-shaped or rectangle-shaped cell next to another one, we can have these junctions along the sides. So those are the lateral sides, as well as, if you remember from anatomy, the bottom of your cells will always have, of epithelial cells, will always have a basement membrane. So that basal surface will also have tight junctions adhering it to the basement membrane. Your book calls the membranes here basolateral membrane. So the membrane that's in light blue here. Let me just erase some of my red marking. So notice that the basolateral membrane is the blue. So lateral, basal, lateral, right? That's our basolateral membrane. All right, so that is our tight junction, so made to really form a tight seal. And last we have something called a desmosome. Desmosomes are filamentous junctions, so we can see we have these filaments that are expanding out into the cell farther away from that membrane, and these are going to be found in places with some kind of mechanical stress. So if you're going to be pushing or pulling or stretching this organ, you're going to have these fibers, and it just kind of anchors and reinforces. So it anchors the junction further out into the connective tissue farther away. So it sort of makes it stronger in terms of like being resistant to some stress. So I have examples like your uterus, the skin, and the heart. They have desmosomes because all those organs are, especially the uterus during childbirth is under tremendous amount of pressure and stress. All right. So last image here of your organelles is the protein synthesis, or just a quick overview of how all your organelles work. So this is to recap, right? So your nucleus contains your DNA. We have our nuclear pores where RNA is exiting. We have the protein being made on a small little ribosome. Ribosomes can appear on the rough ER. You notice that the newly made proteins are being put into a outcropping of the membrane. This is going to be called a little vesicle, right? So this transport vesicle buds off of the ER. This transport vesicle will move to the Golgi apparatus, where it's going to fuse with the cis face of the Golgi, move through the Golgi apparatus. The Golgi will modify and package up those proteins into another vesicle, okay? And this vesicle will transport the substances out of the cell. So if the vesicle is fated to go out of the cell to secrete substances, it's just called a secretory or secretory vesicle. It could also, right, remember that the membrane itself has proteins. So it could be that the proteins that are made in the rough ER are basically receptor proteins or channel proteins that the cell membrane needs more of. So this vesicle can fuse with the plasma membrane, and you can see the proteins remain embedded in the plasma membrane, and that's how the cell can renew the receptors or make more receptors or channel proteins in the plasma membrane. It puts it up there in this way. And then another job of the Golgi apparatus is to make a lysosome. So we have another pathway shown here where we have a lysosomes made. So what's inside of this vesicle would be your digestive enzymes and acid. All right, so that's just a quick recap of how molecules would flow through, newly made proteins would flow through the cell. Now, moving on to the cell cycle. This is just a quick review of mitosis and meiosis. Actually, I'm not going to go over meiosis, just mitosis. So the cell cycle is important to understand in terms of cancer. Cancer is a huge disease that we have in society, and it is very complicated. There's many kinds of cancer. Each cancer has sort of a different problem that arises, so that's why it's very difficult to get like one silver bullet to cure all cancers, because every cancer is so different. But overall, cancer is a disorder in the cell cycle. So there's something not working in the cell cycle. It's disrupted and it continues on and on and on, unchecked. But to understand the cell cycle, let's just remember that the cell cycle includes interphase. Interphase is a very long phase. It's the longest phase the cell is in during the cell cycle. The G1 is the gap 1 phase, where the cell is growing and doing its regular job. The S phase is where the DNA is actually duplicated, right? The S stands for synthesis, for DNA synthesis. In the gap 2 phase, the cell is continuing to grow to prepare for mitosis. When we reach mitosis, this is active cell division, so the cell is now dividing. Mitosis has 5 subphases. We have prophase, metaphase, anaphase, telophase. Prometaphase is, some teachers might teach that there is a prometaphase. So it's between the prophase and metaphase stage. Some people call it late prophase. And then telophase and cytokinesis kind of go together, right? So through these mitotic phases, the chromosomes are actively moving to opposite ends of the cell. The sister chromatids are separating. And then cytokinesis is the cytoplasm breaking in half, right? I want to point out here that you can exit the cell cycle. So some cells don't reproduce anymore. The most common cell you've probably heard of that they don't replace themselves are neurons. They exit the cell cycle and then their resting phase called G0. Okay, so the G0 phase means that they no longer reproduce themselves. They are in this resting phase. Can we get neurons to re-enter the cell cycle, right? People are working on that. But cells that regularly divide, like epithelial cells, the lining of your digestive tract, your skin, are constantly going through the cell cycle. So here are some events in each phase: prophase, prometaphase, the late stage of prophase, metaphase, anaphase, telophase, and cytokinesis. And then here is a nice image of a ball of cells. So each one of these structures is one cell. Okay, so how do we get here? Through mitosis, right? So this is a developing embryo. And this ball of cells is through many, many cell cycles. One cell divides to two, two to four, four to eight, etc. All right, so let's look at your table in your, let me go back here. To look at your table in your outline, this is basically what you should also know when you compare mitosis and meiosis in terms of the function and the overall output of each process. All right, and that concludes this chapter.